First, I would like to thank the organizing committee for the kind invitation and the opportunity to share in this third electronic conference on medicinal chemistry this topic of old drugs with new applications, in which I will present two case studies of old drugs, glucantone and mitoxantrone. My name is Esousa. I'm a professor of medicinal chemistry at the Faculty of Pharmacy, University of Portugal, and a researcher of CIMAR. In this graphical abstract, I want to highlight that we use glucantone as a model to pursue inhibitors of P-glycoprotein with anti-tumor activity and the ability to revert multi-drug resistance, and for that purpose we followed a structure-based design. In the second case study, we wanted to answer the question: are mitoxantrone metabolites responsible for their cardiotoxicity? And for that, we synthesized known metabolites of this anti-cancer drug to investigate their cardiotoxic effects. And this is the abstract of my talk. I take the opportunity to share this invitation to this special issue of old pharmaceuticals with new applications, active on Pharmaceutical Journal, and it's close related with the topic of my talk. So in recent years, we have experienced a surge of interest in drug repurposing. This is a trend finding new uses for existing drugs, especially in diseases where there is an unmet clinical need, such as neglected and orphan diseases. Other opportunity is the development of novel applications using combinations of all drugs. So this famous statement from Sir James Black, that the most fruitful basis for discovery of a new drug is to start with an old drug, has received many adherents this century, not only in finding new applications, but also looking for the unexploited potential of old drugs as starting points for molecular modifications. The Journal of Pharmaceuticals has now opened a special issue and invites both reviews and original articles shedding light on the challenges and opportunities of using old pharmaceuticals in drug discovery. So the first case study of glucantone, that is a thioxantone, that was introduced in therapy in 1945. Although it was considered at the time a drug of choice as anti-schistosomal agent, in the 70s it was withdrawn from therapy due to mutagenic side effects. Nevertheless, this effect led to the investigation of this compound as an anti-tumor agent. Concerning its mechanism of action, it is a DNA intercalator and inhibitor of the DNA repair enzyme apurinic endonuclease 1. It is orally available and sensitizes tumor cells to radiation and chemotherapy, entering clinical trials with that purpose. Nevertheless, some cardiotoxic effects were noted for this drug, and studying several generations of these compounds, these cardiotoxic effects were ascribed to the presence of a methyl or a methylene unit in this position 4. So in our project, we designed compounds that could eliminate in this position the methylene group and introduce molecular descriptors that could produce P-glycoprotein inhibitors. The idea is that an anti-cancer drug having a PGP inhibitory effect could be more effective in overcoming resistance. Following a structure-based design, we were able to design and synthesize a library of 30 compounds. First, we investigate these compounds as inhibitors of tumor cell growth, and we achieve a set of nice results with 6 derivatives with potent effects while not affecting the growth of non-tumor cell lines and also achieving in vivo efficacy. The cellular mechanisms of action were also investigated and include apoptosis and involvement in the cell cycle, and also we found an autophagy modulatory effect to be associated to our EAT compound. When investigating their effect on the rhodamine 1,2,3 accumulation assay, we found half of the compounds to be PGP inhibitors. What could be seen as quite disappointing was the fact that our docking model could not distinguish between an activation and an inhibition effect, and also half of the compounds decreased the rhodamine accumulation. But these results could be very interesting from a toxicological point of view, and we discovered in further studies that these activators could protect cells from pesticides intoxications. In summary, this old drug was a promising starting point to the discovery of potent anti-tumor and PGP inhibitor dual agents, and new mechanisms and new applications were disclosed for some of these derivatives. Entering the second case study of mitoxantrone, this drug is an example of drug repurposing. It was approved in 1987 as an anti-tumor drug and in 2002 for use in multiple sclerosis. Nevertheless, mitoxantrone shows a high incidence of cardiotoxicity, which limits mitoxantrone dose in patients receiving intense or prolonged therapy. Still, the underlying mechanisms of mitoxantrone cardiotoxicity are poorly understood. Therefore, the aim was to synthesize drug metabolites to study their toxicity, since the metabolism of several anticancer drugs seems to have an important role in their cardiotoxicity, such is the example of doxorubicin. So first, we need to have this global picture of mitoxantrone metabolic pathway. And although carboxylic acid derivatives were the major metabolites, we decided to start with this naphthoquinazoline metabolite that was already described as an active metabolite. The synthesis followed a biomimetic approach using horseradish peroxidase. Here I just would like to highlight the difficulty in purifying such similar compounds, so we need to perform HPLC for this purification and establish its structure correctly. Also, we were concerned with a high purity in this synthetic metabolite. We then want to compare its cytotoxicity to the parent drug to determine whether this metabolite is or not a major contributor for mitoxantrone cardiotoxicity. And we performed that in a cardiac model in vitro. I'm not going to enter in details, but generally mitoxantrone was more toxic in all cytotoxic tests performed. Moreover, we understood that autophagy was involved in the toxicity of both compounds, and energetic pathways were affected in a different manner. What I want to highlight here is that this was the first broad study on naphthoquinoxaline cardiotoxicity and revealed that the parent drug, mitoxantrone, caused a higher disruption in the energetic pathways in a cardiac model in vitro. Since naphthol was claimed to largely contribute to mitoxantrone anti-cancer properties, we propose this metabolite should be regarded as a good option for a safer anti-cancer therapy, being less cardiotoxic than mitoxantrone. We are now continuing with the synthesis of carboxylated derivatives. As you can see, it follows a classical chemistry, but even though we have quite tricky steps, for instance, it was very hard for us to get this regioselectivity in the nitration of chrysazine. Also, we performed the synthesis of acetylated derivatives. With this case study, I would like to finish giving the example of the opportunities that are emerging in this case of studying metabolites of old drugs, and not so old drugs such as the kinase inhibitors, since their increasing use is followed by an increased risk of cardiotoxicity. For instance, sorafenib is a multi-target kinase inhibitor and is one of the most cardiotoxic drugs. And already we have 8 metabolites being identified. Nonetheless, no assessment of the potential cardiotoxicity of these metabolites was done so far to the best of our knowledge. So in conclusion, with these two case studies, and what I want to show is that using whole drugs as models in this drug discovery process has quite appealing advantage, such as the quality of our starting points. Of course, you can criticize by the lack of innovation, but I hope that I could convince you in this talk that these two drugs open several pathways and show us many directions, new directions that we can follow. Finally, I would like to thank the entities for the financial support and hope that you enjoy such an exciting conference.